So I would first just want to start out thanking everyone who came, obviously, either here in person or on the Teams call. I'm John Kaufman, and I'm a bio major, and my presentation and capstone project was on the effect of food availability on the wingless trait in fruit flies. Now, whenever you do a capstone project or anything like this, the first question is always like, why would you do this? Why does this matter? Now, my first big thing is evolution. It's something that's always interested me, and it's one of my big reasons for wanting to become a bio major. And there was a recent study done by the Pew Research Foundation, which found that only about 60% of of Americans believe that evolution is real and that it is affecting our daily lives, while about 97% of the scientific community believes this. Now, obviously, that's a pretty big disparity, especially for something that is, at least as far as I'm concerned, a pretty basic foundational thing when it comes into biology. So what I wanted to do was create an experiment that can show evolution in work, that is efficient, quick, and almost anyone could do it and find results. So I wanted to use something like an organism that you could do that with, and so something like a dog would be too big because it would take too long to see evolution happening, and also it's usually considered inhumane to have them die off through natural selection. And then you have something like bacteria, which is extremely efficient, but at the same time it does not have that same effect of seeing it like an actual trait being passed down. People just usually aren't wowed by bacteria. And another thing that I really wanted to do was make it practical, something that you can do it anywhere. And so we go to the setup. You have to do this in a lab setting. This had to be done in a lab setting because when you want to make sure that the only thing that's changing here is the fact that it's your factor, which for me was the fruit availability. And what I did was I put them in a little container like this. Well, this was the container actually. There's still some dead flies in here. And what you would do is put a mesh over the top, and then in one container you'd have the fruit on the ground, and another taped to the mesh on top. And then slicken up the sides of it with some Vaseline to make it extremely harder for the flies to climb up. And so that would put a pressure on them that a wing trait would be very helpful. And another thing was that for the genes, I know I wanted to do winged because in the wing trait is an autosomal recessive gene. And what that means is that it's only one effect. It's not sex linked, so that means that their gender does not affect it. All you have to look at is whether or not they have wings, and you'll know exactly what the genotype is. And then also something that someone in this class brought up, Noah, was what would I do if every single fly had died during the test and I had to change this, was I had kept a small breeding population within my tube so that after a few weeks, this would have a whole nother breeding population that I could put back in and see and restart it if I had to. So, and then as I said before, the differences was that one would have the fruit stuck on the ceiling, and so with the slickened up sides, which would make it an extremely difficult pressure for anything without wings to get to it, because they'd have to climb through the Vaseline. And then the other one had the fruit on the ground, which made it a lot easier, obviously, and there was no pressure to have wings. You could just crawl right to it. And now for the data gathering. There's multiple ways to try and count the flies and see the traits on them. I chose to go with something I had done before in high school, which was to chill them and knock them out. And it's an efficient and easy way, which is one of the big reasons why I wanted to do it. And the other thing, but there are some faults with it, stuff like the overlap between where some get knocked out and some die is pretty large. So there were times where the last one to knock out would lead to one or two dying in the test. I would use a dissecting scope instead of something like a microscope, just because a dissecting scope would let me see large groups of them underneath it, while a microscope would be too thin. I wouldn't be able to see enough, and the amount of time it takes for them to wake up, it would take way too long. And then my data would be analyzed with a chi-square analysis, because I wanted to make sure that the populations and the way I was analyzing them was actually showing a trend, and that this data actually meant something. That this wasn't just random chance that more were wingless and more were winged. And then I was going to do this every week for 6 weeks because, again, I wanted this to be efficient and something that most people could do and see the results, with the first week just being my initial count of when they first came in. And now for my expectations, I thought there would be more wingless in box 2, which is the one with the fruit on the ground, because obviously there's no pressure to stop it, and I wanted to see adaptation shown quickly. Now, one thing that I did expect was that, is that in the box 2 with the fruit on the ground, that the wingless trait would stay consistent, that it wouldn't be increasing or decreasing just because there is no real pressure. But if I go to my chart, what I really want to focus on is the control of the wingless and the experimental, because these are the actual numbers, and then the proportion counts. Now, when you look, originally I started with about 12% wingless here. And then in the experimental, I was about 19-20% wingless. And then as you go down, like especially in the control, this really stood out to me, and it's something I did not expect, was that the wingless population started to skyrocket around week 3. It went from 13 to 30, and I did not expect that because there was no pressure making them go wingless. The only thing that I was able to come up with, and there was actually a study about, or I found an article on this on Nat Geo, which is about, there was like this case where animals would fall into holes and die, and they would consistently keep going down, and the flies that would harvest and like subsist off of these corpses would then lose their wings because there's such a steady supply of food that they do not need to leave anymore. And it's just a waste of energy almost to have these wings. It does not help them anymore. And so I thought that was interesting because I did not expect that to come about so quickly. It was only 3 weeks. And then in the experimental group, you can see that the numbers start to drop very consistently here. Like it starts at a 12, and then 5, and then 2, and then it just kind of stays in that single-digit range. And the explanation for that was that was something I had expected, was because that trait is always going to stay there because since it's autosomal recessive, like I said, there's always going to be a few winged parents that are heterozygous, so they are dominant for the winged, but then still have that trait, and they'll mate, and there'll be a few that'll come up and they'll die off because they're going to struggle to get to the food. And then obviously you can see, and I also kept track of the flies that I had passed just because I thought it was important to see how many I was killing, whether it was because of I overfroze them. Sometimes they woke up a little bit faster than I had expected, and so they started to run away, and I would just have to smash them because I'm not letting them run around. And so that was something I really wanted to keep track of. And then here, like, this is the chart that I had. The blue is the control, the orange is the experimental, and you can see like just the steady drop in the experimental of the wingless. And then you can see on the blue how it just skyrockets around week 3, up to over 50% of them being wingless, even though like your expected would be about 25%. And then here's my chi-square value, which I mean the big thing here is just that every number that I got was substantial, that it meant something. This wasn't just random chance. And then here for the discussion, the continued presence of wingless, like I said, was a big thing that was important to see that it's still there, because that also plays into the evolution talk, that these traits are still there and they still kind of stay around just as almost like a backup, but they're always there. And then the increase of the wingless was something that was really crazy to me again. And here you can even see what they look like, because there's different wing traits that can pop up. Like you have the dumpy wings and the miniature, and then true wild type. Both of these would be considered flightless just because miniature is too small for them to carry themselves up. And then again, the chi-square, every single number that I got was significant. There was never a point in it where these were just, where the chi-square said this was just random chance. And then for the applications, the results were fast. I was very satisfied with how quickly things came around. And then it's an easy setup. Like most of this stuff you can find in your own house, other than the flies, which only cost like $20 if you get them on Carolina.com. So that's a really easy thing to come by. And like I said, it's very cheap, very easy to get. And that is it.